welcome everyone. Welcome back. Uh, Today we're doing chapter 12, and this is going to cover basically the cell cycle, and we're going to cover how the cell divides, which is mainly called mitosis. Um, cell cycle is basically the life of a cell. You know, the cell has like a lifespan that basically it grows and matures in, uh, and then eventually it divides to become two cells instead of one, and two cells become four cells, and so they keep doubling. Uh, you know, that's the main thing of cell division. Okay, so this is a kind of a depiction of how cells, you know, divide from like one cell and then they basically become two cells and then they become four cells. You see that? So just right off the bat, just some things to remember are the nucleus of a cell has the cell's DNA inside it, right? And so whenever we want to divide a cell, you know, we want to make sure that the same genetic information that's in the DNA is basically copied and pasted to the next um, offspring daughter cells. Okay, so otherwise, you know, you have variations, and in mitosis or the regular cell division, you know, we have the same genetic information being passed on. Okay. So this is a video of sea urchin embryo. So this is just, you know, you can see on your own, showing, you know, how cell division happen, and you can actually like see the, you know, different cells inside the sea urchin, which is pretty cool. Okay, so the rules of cell division. Uh, So it's again the ability of organisms to produce more of their own kind, right? That's what distinguishes living things from non-living matter, right? All living things like plants, animals, you know, including humans, right? Um, we are able to, um, you know, divide our cells, and so we can generate cell daughters, right? Daughter cells from a parental cell, okay? Same way we can also like produce gametes, right? Uh, Gamete cells, and that's a different type of division that, you know, we're not going to talk about it mainly today. We're talking about just mitosis, which is actually the cell itself dividing, okay? We're not talking about gametes here in this chapter, not yet at least, okay? So this is what distinguishes living things. Living things want to reproduce and maintain themselves, okay? Um, so the continuity of life, like I mentioned, is based on the reproduction of cells, or that's cell division. Okay, another word for it is mitosis. Okay, so there are different types of you know, how cells divide, right? Whether it's a unicellular organism or a multicellular organism, right? So in unicellular organism, the division basically of one cell reproduces the entire organism. Okay, so it's a bit different. However, in multicellular eukaryotes, right, such as humans, um, basically they depend on cell division for development from fertilized egg, growth, and repair. Okay, so one cell becomes two cells becomes four cells, right? That's for growth, right? You're growing the number of cells, and so also, like, you know, growth, as you know, we're talking about growing from being a child to you know, to, you know, the different period of age. Okay, so that's growing, and to do that you need cells to divide, Also, whenever we injure, let's say ourselves by, let's say, cutting your finger or something, or maybe like you're playing sports and you fall and you injure your knee, and so that repair process, basically the cells have to divide, um, and basically make more of their same same kind to be able to regenerate the the cells that were damaged. Okay, and that's how the repair happens. Okay, now in meiosis, which is the other kind of division, that's when we have a production of basically gametes, like sperm or an egg. That's for reproduction. Okay, so cell division, again mitosis, is an integral part of the cell cycle. Okay, so cell cycle again is the life of a cell, and then it goes through basically maturity and growth and maintenance, right? And eventually get to the point where it wants to divide and create basically daughter cells based off of it. Okay, so let's see here. Most cell divisions, as I mentioned, result in two daughter cells, and two daughter cells result in four daughter cells, and so on. So that DNA that's in the nucleus is just being passed on to those daughter cells and the same exact DNA. So there's no variation in it. Okay. So in meiosis, which is the division that basically produces sex gametes. Okay. Um, this is where you have a sperm and an egg being produced. Okay. Okay, so, and as we mentioned, the cell division is very accurate in passing DNA from one generation to the next. Okay, so this is just showing you how an asexual reproduction happens here. You know, these are also like um, just cells dividing, and these are basically like reasons why they would divide. To have, like I mentioned, growth and development, right? To have tissue renewal in case of like an injury or disease or something like that. Okay. So let's see here. So as far as the DNA that's contained inside the nucleus of a cell, that's what constitutes the cell's genome. That's what we call it genome. Okay, which is basically the full set of genetic information of the whole organism is actually inside that tiny DNA. So each cell has the whole genetic information in it. Okay. So let's see here. Genome can consist of a single DNA molecule, right? Common in prokaryotic cells, or a number of DNA molecules common in eukaryotic cells. Okay. DNA molecules in a cell are packaged into chromosomes. Okay, so um, this is like depicted here in this photo, right here. So you see that spaghetti-like form in blue. So this is basically the DNA. Okay, and in order for it to be like for the cell to divide and and become two cells, those DNA you know zigzags, if you want to call it, they basically condense into chromosomal forms. Okay, so have you heard of the chromosome? I'm hoping. So the chromosomes basically contain, you know, that genetic information inside them. So basically, us humans, right? We have like half of our chromosomes, total chromosomes, come from basically like 46. Okay, so 46 chromosomes, half of them come from, you know, the our father, and half of them come from the mother. Okay, so when they combine, they give you the 46 chromosomes. Okay, sorry about that. Okay, So the DNA molecule of a chromosome, right, carries several hundred to a few thousand genes. So as I mentioned, so the DNA, when we talk about DNA, we're talking about the genes, right? What do genes do? They carry genetic information. That's the coding, okay, to that code for development and for maintenance and for the whole thing basically, and how everything works together and how everything basically gets created. Okay. So eukaryotic chromosomes consist of chromatin. So chromatin is basically a complex of DNA and protein that condenses during cell division, right? So if you look at I don't have I should have put a chromosome here to show you. But a chromosome, you can look it up and see how it is, right? So it's got basically condensed, as I mentioned. So that DNA is condensed into it, okay? And once you unwind that DNA, okay, you'll find that this DNA is like basically is turning around what we call histones, okay? So these are proteins, and together the DNA and the protein basically form the that complex form your chromatin, okay? So so every eukaryotic species has a characteristic number of chromosomes in each cell nucleus, right? Um, so we have two types of cells. We have somatic cells, okay? So any cell that basically exclude, the um, reproduction cell or sex cells, then these would be like, we call them somatic. So like skin cells, muscle cells, nerve cells, anything you wanna, you can call besides sex cells or reproduction cells, we call them somatic cells. Okay, so these have two sets of chromosomes. Okay, so basically these will have two sets of chromosomes, half of them from the mother and half from the father. Okay, gametes. Okay, on the other hand, when we say gametes, we're talking about reproductive cells like sperm and eggs. Okay, just remember, gametes are talking about reproductive cells or sex cells. Okay, have half as many chromosomes as somatic cells. So when meiosis happens, which meiosis produces gametes. Okay, when meiosis happens, we end up with half of the amount of chromosome that we began. Okay, and the reason is because you want to have half of the chromosomes in the sperm and half in an egg. Okay, so the reason is because um, when you know when reproductions need to happen, right? You have like a sperm uh, that combines with an egg, okay, and you have basically half of the amount of chromosomes in the sperm and half of the amount of chromosomes in the egg, and that gives you basically the whole amount of chromosomes, which is what, 46. Okay, so this is the reason why gametes have half of the amount of chromosomes. It's because they want to combine with another gamete, which is basically a sperm combines with an egg, and an egg combines with a sperm. Okay, to create a full amount of the number of chromosomes. Okay, so as I mentioned, in the cell cycle, right, the cell basically matures and grow and maintain itself. However, it also has a kind of like a regulation signaling where it knows that it's gonna divide eventually. Okay, it's gonna go through mitosis. So for that to happen, what do we need to do? We said we need to have the same DNA. Sorry about that. So let me mute that. Okay, so we wanna basically have the same DNA, okay, that once one cell has the parental cell, we want to have it in the daughter cells, okay? So in order to do that, what are we going to do? We need to copy paste the DNA or replicate the DNA, okay? And so this is what the cell goes through. So it's called the S phase. We will learn about all the phases, but in the S phase, the DNA of that cell that's contained in the nucleus gets replicated, okay? And the chromosomes condense after that, okay? Now, we also want to learn about an area, right? Um, basically, inside the chromosome, and it's called centromere, okay? Um, so that area is a narrow waist of a duplicated chromosome where the two chromatids are most closely attached, okay? So, um, let me see. Okay, so here's that. Let me just show you that before we move on. So this is basically what we call a chromosome, okay? By itself, we call it a chromosome. Whenever it becomes two, we call them sister chromatids, okay? Now in the middle, right here, do you see that? Where basically the short arm and the long arm of a chromosome meet, we call this centromere, okay? So let's continue this, okay? So each duplicated chromosome has two sister chromatids, as I mentioned, okay? You see that? Joint copies of the original chromosome. So, after this gets duplicated through what? Through the S phase, which is basically where the DNA is replicated, okay? We get basically two sister chromatids. You see that? Then, okay, after that, basically the cell is ready for cell division to happen. Okay, we want to basically replicate the DNA to get it ready. Okay, so during cell division, okay, the two sister chromatids of each duplicated chromosome separate and move into two nuclei. Okay, so this is the process where we have basically those duplicated sister chromatids that have the DNA right contained in each. They separate, and we will see how that happens during mitosis, and they move into each of its respective uh, nucleus of each newly formed daughter cell. Okay. So this is showing you also the sister chromatids under the microscope. So you're looking at basically a sister chromatid here and another one here, and that middle part is the centromere. Okay. So we said that once they're separated, each one of these we don't call anymore sister chromatid because it has separated successfully, so we call it chromosome at this point, okay? So, as you can see here, we explained this already, okay? Watch this. So, now let's talk about the parts that happen in a cell division, eukaryotic cell division, okay? So there are two parts. We have the mitosis, which is basically the division of the genetic material, which is the chromosomes. So remember, we've duplicated the chromosomes, or the DNA, right, in the S phase of the cell cycle. Now, when we're ready to divide the whole thing, we are going to start with mitosis. Mitosis is specifically the division of genetic material, okay, which is contained where? Inside the nucleus. Okay, so you might be asking, like, what about the actual cell itself? Okay, don't we need to divide it and like maybe copy, the organelles and everything, basically make exactly the same cell, but two of that cell? Yes, so this is where cytokinesis, that's the division of the cytoplasm. Do you remember cytoplasm? Cytoplasm is basically the whole thing that basically is around the nucleus of a cell. Okay? Now, it's just cytokinesis is exactly, comes from the word cyto, which is cell, kinesis, division. Okay? So it's basically dividing that cell completely and making two cells. Okay? So gametes are produced by a variation of cell division called meiosis. So while somatic cells, regular cells, okay, like skin cells for example, are going through mitosis, which is regular cell division, okay, in order to produce gametes, which do you remember what that is? That these would be what? The sex cells, okay, reproduction cells. So these would be like sperm and egg, okay. In order to produce those, you want to have meiosis. You want to go through meiosis. So meiosis produces variations in the cell. So it's not exactly the same like mitosis. Mitosis, you're copying, basically copy paste the DNA, the same thing. However, in meiosis you have variations, and when we study that we'll get to it. We'll explain more of it. Okay. So again, meiosis yields non-identical daughter cells that have half as many chromosomes as the parent cell. So we already explained this. Okay? Alright, so let's take a look a little bit at the cell cycle here. Okay, so, and explain the different parts of that cell cycle. Okay, so the whole thing here is represented by just that cell cycle is that circle, 360-degree circle. Okay, so the cell basically start out from here. So you can see it's got an unduplicated chromosomes. Okay, and this phase, by the way, all of these parts we call them interphase. Okay, during interphase we have what we call the maintenance and cell growth. Okay, that's the one that precedes mitosis, which is this is the only part where cell division happens. Do you see it? Okay, so interphase, again, you have all of these G1 phase, you have the S phase, that's the one I mentioned where DNA is replicated. Okay, then you have G2 phase, all of these we call them interphase. Okay, now in mitosis or cell division, this is where we have the genetic information gets divided, okay, which has happened in mitosis. And this is where we also have at the end cytokinesis, where basically the actual whole cell divides and become two. Okay. All right. So let's go ahead and keep explaining this a little bit. So interphase is 90% of that cell cycle. Okay. You can see how the cell basically is like maturing and growing and dividing its organelles and basically replicating its DNA in the S phase, doing all of these 90% of the time until it gets to the mitosis part. Okay, so G1 phase, you have metabolic activity and growth, that's where the cell starts. Okay, call it first gap. Then you have the S phase after that. This is where you get your the DNA basically gets duplicated. Okay, so those chromosomes get duplicated. Now you have like two copies. Okay, and then basically in the G2 phase, the chromosomes starts like they want to condense into their chromosomal shapes. Okay, and the cell basically have already duplicated its organelles here, and it's ready for cell division. Okay. Okay, this is showing the same thing. Alright, so now we will see how the cell division actually happens. Okay, so we have basically different stages that mitosis happen in. Okay, so at the end of G2 phase of interphase, okay, so we're right here basically. Okay, so the cell has again matured and basically grew, right, and replicated its DNA, replicated its organelles. Let's see how that looks like. Okay, so this is your cell. We said that it's got already duplicated chromosomes. Do you see that? They're uncondensed yet, not yet. Okay, so they're gonna start condensing to take on their chromosomal shape. Okay, as I mentioned. And then you have your centrosomes, which are also duplicated. We will see why the centrosomes are actually uh, important, right? What they do. Okay. And you have your nucleus here that's containing, your chromosomes. Okay. And you have the nucleolus here, which is a small organelle here. Okay, and you have your nuclear envelope, which is basically encompassing and containing you know, the um, duplicated chromosomes inside. Now, so let's look at prophase. That's the first phase of mitosis. Okay, so that spaghetti-like DNA basically wants to take on the condensed form of chromosomes, and it does that because eventually during cell division we want to separate those um, chromosomes, which are made out of what? Two sister chromatids. Okay, so we want to separate it, and to do that, they have to take on that shape to be able to be separated, at least correctly. Okay, so we can also notice that this nuclear envelope that was full here previously, it starts breaking down. Do you see that? Now you might ask, why do we need to break down the nuclear envelope? Well, we need to because we want to get this chromosomes, genetic information, out of the cell to be able to actually divide that cell. Okay, think about the nuclear envelope as like a border that's like not allowing division to happen. So we need to basically start disintegrating that border to be able to get the genetic information out of it. Okay. So so the centrosomes now, okay, that were duplicated previously, they migrate towards the two poles of the cell, so the north and the south pole. Okay, there's a reason why they do that. Okay, now you will notice that there are little like uh, rope-like structures coming out of those centrosomes, and these are what we call microtubules. Okay, so these microtubules are exactly like um, really like cables. Okay, and they will act like cables eventually. Okay, you will see how that happens. So they will start moving out of the centrosomes. Okay, they will move towards each other, as you can see here. They will also move towards the centromere, which is that middle part of those chromosomes that we talked about, right? Okay. So after that happens, okay, we move on to the next phase, which is metaphase. Okay. So what happened in let me tell you something before we move on. So in order to remember the order of these phases, remember the word P mat. P mat. So that's each letter corresponds to the first letter. So P for prophase, M for metaphase, A for anaphase, and then T for telophase. Okay? So this will help you remember the order of it. Okay, so now we're at metaphase. Now also I want you to remember one more thing, okay, which will help you in a test question. Okay, so if I ask you in which phase do those chromosomes actually line up in the middle, so you're going to answer metaphase. Okay, so think about meta is middle. Okay, so as you can see here, you can notice that the microtubules, so those cable-like structures, right? When we studied about these, right, and they're part of the cytoskeleton of the cell. Okay. So you can see how basically they're completely now attaching to the centromeres of chromosomes and to each other as well. Okay? And you can notice that the nuclear membrane that was once around those chromosomes is completely gone now. Okay? There's no more. Now, what happened in anaphase? So that would be your third phase. During anaphase, right, the microtubules, which are these cables, they actually pull those sister chromatids away. Do you see that? Hopefully this is making sense now, right? Those sister chromatids were basically attached with each other, right? And forming that, each chromosome, okay? So when the microtubules attach to the centromere of chromosomes, they're able to pull on those sister chromatids. And by the way, so the centromere is the location of the chromosome, which is the middle part between the long arm and the short arm. However, on that location, we have a protein complex, and it's called kinetochore. Okay, that kinetochore is basically where the microtubules actually attach. Okay, so that section right here, which is so if I, the way I think about it is, kinetochore are located at the centromeres of a chromosome. Okay, so hopefully this is clear. So they attach to the kinetochore at the centromere, and they pull those sister chromatids toward the north pole and the south pole of the cell. And they're doing that because, they want to separate that cell and divide it into two cells eventually. So you can see how the genetic information is basically moving towards the north pole and the south pole. Now during telophase, okay, now chromosomes are basically unwinding again here. So after they were condensed, and prophase, right, in the first phase, okay, so now, division is almost being completed, right? They do not need to be in their condensed form anymore. So you get that spaghetti form again, okay? The nuclear membrane now starts appearing again. You see that? Why? Because now we want to contain those uncondensed form of chromosomes, okay? We want to put them inside the nucleus only. We don't want the genetic information to be just like flowing and floating in a cytoplasm of a cell. Okay, and basically we're redoing what was undone in prophase. Okay, now the final stage of mitosis or of the cell division is cytokinesis. And again, cytokinesis is, the division of the actual, cell into two cells and becoming two daughter cells. Okay? So you see they're dividing at that cytoplasm. Okay? You can watch that, watch that, watch that. We're not gonna basically take the time to do that lecture portion. Okay? So let's take a look at the mitotic spindle, okay? The mitotic spindle is a structure basically made of microtubules. So those cable-like that we were looking at, right? These control chromosome movement during mitosis. Okay? So again, you can see how those microtubules were able to pull the sister chromatids toward the north and the south pole. Okay? In animal cells, assembly of spindle microtubules begin in the centromere. We saw that, right? We saw how those centromeres, right? Those orange structures here, we saw how they were able to form those microtubules and basically get them out of them to be able to pull on the sister chromatids. Okay. So the centromere replicates during interphase, right? When we said that all the organelles are replicating during the interphase, okay, as well as the DNA being replicated in the S phase, okay, which is part of the interphase. So again, those centrosomes are migrating towards the opposite ends of the cell, okay, during prophase, as we saw, okay. Now, an ester, okay, extends from each centrosome. So the ester would be these short microtubules right before they actually starts extending all the way to attach to the other centromere, not centromere but centrosome, sorry, and also to centromeres of chromosomes. So these pack right here, that's what we call ester. Okay. So the spindle includes centrosomes, the spindle microtubules, and the esters. So this is talking about what considers the whole system. Okay. So that's basically explaining kind of what we explained a little bit earlier. Okay. I'm just like repeating this stuff so you guys can understand it a bit more. Okay. So each sister chromatid, right, looking at these, they have a kinetochore. Where does a kinetochore, where is it located at? It's at the centromere of a chromosome, which is right here in the middle. So it's a protein complex associated with centromeres. See that? During pro-metaphase, which is like, right before the actual metaphase, some spindle microtubules, right, kinetochore microtubules attached to kinetochores. Okay, and we said that. Okay, so they start basically forming in prophase, but they attach completely, during metaphase, right? And where happened during metaphase? The lining up of chromosomes in the middle. Okay, that middle we call it an imaginary plate, which is called metaphase plate. Okay. Okay, so let's talk a little bit more about the mitotic spindle, right? So let's see here. Okay, so looking at the in metaphase, right, when we have um, the lining up of chromosomes, right? Now, how do actually the microtubules, okay, pull on those sister chromatids towards the two poles of the cell or towards the centrosomes? How do they do that? So the way they do it is they pull, yes, but they also, they're like rope, and they climb. Think about them as climbing um, towards the other end. Okay. So, but before they do that, right? Let's talk a little bit about cohesions. Okay. So in anaphase, remember what happened in anaphase. Anaphase, we've already, this is what happened right here. We've already separated the sister chromatids and we're pulling them towards the poles. Okay. So the cohesions are cleaved by an enzyme called separase. Okay. So the cohesions, if you want to look at them here, are actually this pink part. Okay, right here. And what they do is they're like sticky part. Okay, so separase is as an enzyme that basically breaks apart cohesion. Okay, so when that happens, sister chromatids separate, okay, and move along the kinetochore microtubules toward the opposite ends of the cell. Okay, so how does that happen again? The microtubules shorten by depolymerizing at their kinetochore ends. Okay, So they're really shortening those microtubules by depolymerization. We will see how that happens in a bit. So, the results of a pretty cool experiment that scientists did was suggest that motor proteins actually they're located on the kinetochores. Kinetochores, remember, they're that protein complex located where? Right here, where those microtubules are are attaching. Okay. So that experiment suggests that those motor proteins on kinetochores actually walk the chromosomes along the microtubules during anaphase. So these chromosomes actually, the experiment suggests that those protein allow that chromosome to walk towards that pole. Okay. The depolymerization of the microtubule at the kinetochore ends right, occurs after the motor proteins have passed. We will see a little depiction of what I'm talking about here. Okay. So this is called a Pac-Man mechanism. So you know how Pac-Man looks like, right? So it's kind of like moving and eating along. Okay. So let's take a look a little bit here at this experiment. So looking at, what is that? That's anaphase, right? So why do we know this is anaphase? Because you can see the sister chromatids have already separated. Okay. So let's look at the experiment right here. So you have the centrosomes, right, on each end. Okay. And you have the kinetochore, which is right here in the middle of the sister chromatids. Okay. Same information. That's nothing new than what we were just saying, a few slides ago. Okay. And so the scientists marked an area in the microtubules right here. So they marked it darker color. Okay. And what they saw was the result. They saw that the sister chromatids were actually closer towards that marked area. So they noticed that the microtubules have shortened. Okay. At the same time, they noticed that the sister chromatids have actually walked towards this end. Okay. So this is the when we say the Pac-Man mechanism and depolymerization, this is what we're talking about. So if you take a look at the the kinetochore, which is located where? Right here. You see that? So think about this as a zoomed in of that sister chromatid. Okay. Okay, you'll see how this microtubule right here is attached to this protein complex called kinetochore. Okay, and on that protein complex we have basically uh, a bunch of like enzymes and so on that basically digest um, this microtubule, and due to this, basically the chromosome, which is this sister chromatid right here, is able to move along this way towards basically the left side. Okay. So other research shows that chromosomes are reeled in by motor proteins at the spindle poles. So again, you have some scientists say that instead of like the microtubules being digested, right, and shortened as a result by depolymerization, we have basically some scientists saying that there are motor proteins, kind of like a someone pulling that rope or microtubule and pulling those sister chromatids only. So it's a motor protein. Okay. So the general consensus of this is we think that both mechanisms are happening. Okay, so let's see here. So non-kinetochore microtubules from opposite poles overlap and push against each other, elongating the cell. Okay. So why would they want to do that? They want to elongate the cell to be able to actually divide it. Okay. So they're pulling that cell, elongating it until eventually we're gonna divide it in the middle. Okay. So at the end of anaphase, duplicate groups of chromosomes, right after we separated those sister chromatids, have arrived at opposite ends of the elongated cell, right? This is the reason why we have microtubules, and this is why we're pulling on those newly formed chromosomes, now we call it, right? So now cytokinesis can begin after we elongate the cell and everything is ready for actual cytoplasm division, okay? So the cells divide and basically we have two cells now, okay? Right before they do that, the cell basically have this cleavage furrow we call. Okay, so the first sign of cleavage is the appearance of a cleavage furrow, as you can see here. Okay, this is showing you that the cells are about to be cleaved off. In the process of cytokinesis and forming your two daughter cells as a result. Okay. Now let's just talk briefly about plant cells. So we talked about animal cells and so on. So in plant cells, a cell plate forms during cytokinesis. So this is a little bit different here. Okay. Do you see that cell plate? And these two are two daughter cells. So there is a cell plate that happens and form in between the newly formed cells. Okay. There are no cleavage furrow as you saw here, like during an animal mitosis. Okay. Just can watch that. Watch that. This is just showing you basically mitosis in a plant cell. Okay. Kind of similar steps here going on, but we're forming that cell plate at the end. Okay. So let's talk a little bit about binary fission bacteria. Okay, so bacteria, which are prokaryotes, right, reproduce by a type of cell division called binary fission. Okay, so in binary fission, the chromosome replicates. Okay, beginning at the origin of replication. We will see how that looks like in a bit. Okay, so I know this does not show, but it's over here. I'll show it to you in a bit. So the chromosome replicates, okay, and the two daughter chromosomes actively move apart, okay. And after that, the plasma membrane pinches inward, dividing the cells into two, okay. So let's take a look at how that happens. So you got basically that's the bacterial cell, okay. You got the chromosome or DNA information here, okay, genetic information. Now you have an origin of replication. That's a starting point right here, shown in the green portion. Okay. So chromosome replication begins, and as a result, you have basically two copies of origin. Do you see that? So instead of basically copying the whole DNA, the way bacteria does, it copies the origin first. Okay, do you remember how in our cells we copy the whole DNA in the S phase? That's DNA replication. Here they're copying basically the origin first. Okay, then after that, one copy of the origin is now at each end of the cell. So after the origin is copied here, it basically migrates to the other end of the cell, as you can see here. Then what happened is the plasma membrane here. Oops. Okay, as we said, it pinches inward, and like you see here, and it divides the two daughter cells. Okay. So because prokaryotes evolved before eukaryotes, mitosis probably evolved from binary fission. Okay, kind of makes sense. So certain unicellular eukaryotes exhibit types of cell division that seem intermediate between binary fission and mitosis. Okay. And so these are just the different types of cell divisions. Like this is a bacterial one that we just went over. This is your most eukaryotes. Okay. And these are some type of yeast and archaea. Okay. So the cell cycle happens as a result of a system regulation. Okay. It always is regulated, but sometimes you have a misregulation in the system and you get problems after that. We will talk about what those problems are. Okay. So the frequency of cell division varies within the type of cell. Okay. These differences result from regulation at the molecular level, right? So they're regulated. Cancer cells, okay, they manage to escape the usual controls on the cell cycle. So this is what you get out of a misregulated, cell division system. Okay. Okay, so we said that the cell cycle is driven by specific chemical signals. Okay. So the sequential events of cell cycle are directed by a distinct cell cycle control system, as I mentioned, right? The cell cycle control system is regulated by both internal and external controls. Okay, so everything in the body, right, is controlled, internally within the cell and externally from due to hormones, due to other signaling molecules that tell things to to start or to stop. Okay, so that control system has basically specific checkpoints. Why do we want to have a checkpoint? We want to have a checkpoint so we can actually check at each step of the cell cycle and see, if that preceding process that the cell went through was actually successful, and we have, a normal cell, okay, and it's not something that's wrong with it, okay. So this is your your cell cycle here, right? And this is showing just how the control system controls the cell cycle. So you have a checkpoints at each of the cell cycle phases. So this would be your G1 checkpoint, okay, G1. It's in the G1 phase, and then you have basically another checkpoint at the end of G2 phase, right before cell goes through mitosis, okay, and then you also have an M checkpoint, which is during mitosis, okay. So there are, as part of this control system, the cells have regulatory proteins, okay. These proteins are involved in the cell cycle. So if these proteins are turned on, switched on, then the cell can proceed with cell division. Okay, so these proteins are called cyclins. Okay, and cyclin-dependent kinases. So abbreviated as CDKS here. Okay. So cyclins are named for their cyclically fluctuating concentrations in the cell. So they fluctuate, and whenever you can imagine, if this is a control system protein, okay, if you have more of these, then the cell will divide. If you don't have a lot of these, then the cell will not divide. Okay. So just know that also the activity of CDKS, which is cyclin-dependent kinases, it falls and rises with cyclins. So they work together. And this is being shown in this table here, or this graph, sorry. Okay. So the way this works is when CDKs attach to cyclin, okay, you get this complex, and it's called MPF complex, as you can see here. Do you see that? So this is the cell cycle that we were talking about. When CDK combines with cyclin, you get the MPF complex. Okay, the MPF complex basically rises and goes down depending on the stage or phase that the cell cycle is going through. So you can see at the M phase, which is what happens during the M phase. Mitosis, the cell division. Both the cyclin concentration and the MPF activity, do you see that? The complex, they increase, and so as a result the cell divides. Okay? That's pretty cool stuff. So they decrease again during G1, S, and G2 phases, as you can see here. Now cancer cells, they do not basically follow the order of these regulatory proteins. Okay? So MPF, that's the maturation promoting factor. So it's a cyclin and CDK complex. Again, when they combine, they form MPF, as I mentioned. That triggers a cell's passage past the G2 checkpoint into the M phase. So this basically tells the cell to divide. Okay, so after it checks off in G2 phase, that means it's good to go. And basically this tells the cell that you're ready to be divided. Okay. So showing exactly the same things. So many signals registered at checkpoints come from cellular surveillance mechanisms within the cell. Okay, so those mechanisms are monitoring, cell constantly, right? If anything goes wrong, they will stop the cell from going through division, and at many cases they might signal the cell to actually die and commit suicide. We're not going to talk about this right now. Okay, so there are three important checkpoints which are in the G1, G2, and M phases, as we saw. Okay. So for many cells, the G1 checkpoint seems to be the most important. So that would be right there, G1. Okay, I can show it to you here. Okay, let's go back to it. I'm sorry, I just want to show it to you. Okay, if a cell receives a go-ahead signal at the G1 checkpoint, then it will go through the S phase, which is right after G1, which is what happened, in DNA synthesis. The DNA synthesis happens in the S phase. Then the cell goes through G2 again to mature and divide its organelles before it goes through the mitosis phase and divide. Okay. So if the cell does not receive the go-ahead signal, it will exit the cycle. Okay. And it switched into the non-dividing state called the G0 phase. And there are actually many cells in the body right now that are in the G0 phase for the rest of their life. And these cells are like neurons, nerve cells. These cells basically, they do not divide. They stay in the G0 phase all the time. Okay. So this is just explaining what we just said right now. Okay. Okay, so this is also explaining the M checkpoint. So the M checkpoint is, what do you think it's going to do? It's inside the mitosis phase. So that means it's checking what? is checking if the cell is actually has actually successfully divided or replicated its DNA. It's the whole thing is working correctly, where the chromosomes are aligning in the middle and doing all of that, right? Um, so without full chromosome attachment, okay, to those microtubules, then the stop signal is received, and then the cell cannot go further to divide and become two. Okay, with full chromosome attachment, as you can see here, uh, right, between metaphase and anaphase, then the go-ahead signal is received and the cell proceeds to divide. Okay, so an example of an internal signal is that cells will not begin anaphase until all chromosomes are properly attached to the spindle, okay, at the metaphase plate. Okay, so this is just explaining what we just said here. Okay, so those chromosomes have to be attached completely by the spindle fibers or microtubules, okay, um, before they can proceed to the anaphase. And because during anaphase, your newly formed chromosomes from the sister chromatids are being pulled apart. Okay. So some external factors, right? We said there are internal factors and external factors that control the cell division. External factors that influence cell division include specific growth factors. Okay, growth factors are released by certain cells and stimulate other cells to divide. Okay, these would be the platelet derived growth factor, so PDGF. Okay, that's one of them. It's made by blood cell fragments called platelets. Okay, so it comes from the word platelets. Okay, what do platelets do? They actually, when you cut your hand, the blood cells that actually seals off initially the area, so no more blood gets out of the skin. And those would be platelets. Okay, so PDGF is required for the division of cultured fibroblasts. Okay, so this growth factor is required for division of fibroblasts. Okay, fibroblasts are a type of cells for now, just know that. Okay, alright, so this is just an experiment the scientists did. Okay, so basically they a sample of human connective tissue, okay, is cut into small pieces, as you can see here, and they put them on a petri dish. Okay, enzymes digest the extracellular matrix of those connective tissue, resulting in the suspension of free fibroblasts as a result, as you can see here. So cells are transferred to cultured vessels, like you see here as well, and that PDGF growth factor is added to half the vessels. Okay, so this one does not have PDGF. This one does. What you notice is that because of the PDGF um, growth factor that we talked about here, those fibroblasts are able to divide. Okay, so this is an external signaling. Okay. Okay, so now there's another external signal that controls cell division is density dependent inhibition. So it's depending on what density. So if there are more cells that are close to each other, cell division is gonna stop. It's gonna basically do a negative feedback. Okay, so most cells also exhibit anchorage dependence to divide. So that means they must be attached to a substratum. Okay, so that means they have to be attached to something around them to be able to divide. Okay, so both types of density dependent inhibition and anchorage dependence check the growth of cells at an optimal density. So they can't have like, you know, too many cells next to each other, otherwise they will stop dividing. Okay, so cancer cells, again, they don't go by any rule. They basically exhibit neither type of regulation of their division. Okay, as you can see here. So, you know, in anchorage dependence, right, cells require a surface for division, as you can see here. Okay, now density dependent inhibition, due to many cells here, uh, you know, the density dependent inhibition control kicks in, and because of that you will see cells, you know, they stop dividing, and then what happened is, you know, cells divide to fill a gap here again. Okay, alright, so let's talk about cancer cells a little bit. So cancer cells do not respond normally to the body's control mechanisms, neither internally or externally. Okay, they do not stop dividing when growth factors are depleted. Cancer cells do not need growth factors to grow and divide. Okay, they make their own growth factors, unfortunately. Okay, they may convey growth factor signal without the presence of the growth factor as well. Okay, so they will do something, you know, as if the growth factor is, you know, is signaling them to do so, but there's no growth factor that orders them. Okay, so they're basically, think about them as like, you know, they do, uh, you know, whatever they want to do. Basically, they're not controlled by anyone. Okay, so they may have an abnormal cell cycle control system. Okay, that control system in this case basically tells them to, instead of like stop dividing, it tells them to divide more. Okay, so there's a malfunction in that cell cycle um, control system. Okay, so cells that acquire the ability to divide indefinitely are undergoing transformation. So these cells basically divide and divide and divide with no stop. Okay, they keep doing it. Unlike a normal cell where, you know, it's got a specific amount of, you know, a number of cell divisions that it will go through before it will eventually die. Okay, so cancer cells, they just go indefinite. Okay, um, their cancer cells are not eliminated by the immune system, uh, form form tumors. Okay, as we know. So if they're left undetected, or the immune system cannot get rid of them, they form tumors, which are masses of abnormal cells within normal tissue. Okay, this is what causes problems as far as like symptoms in people. Okay, if abnormal cells remain only at the original site, so if those, you know, many masses of abnormal cells, if they remain on one specific location of the body, then we call this a benign tumor. Okay, it's a lump. Okay, now most benign tumors do not cause serious problems because they're local. I mean, um, really it depends on, you know, where that benign tumor is. It might eventually cause something because, you know, those tumors basically are occupying space. Okay, and they form a mass, you know, in that space. So you could imagine, you know, that could put pressure on tissue around that uh, masses, and that would cause problems. Okay, so malignant tumors, okay, these are the cancers that actually move around. Okay, so they invade surrounding tissues and can undergo metastasis, which is basically the spread of cancer cells to other parts of the body. So a tumor might start in the, for example, in the intestines, and the tumor might spread from the intestines to, for example, the liver or the brain or other parts of the body. Okay, so that spreading um, mechanism we call metastasis. Okay, and the tumors that actually spread are called malignant tumors. They're not benign. Okay. Now, localized tumors may be treated with high energy radiation, right? It's radiation therapy or chemotherapies. Chemotherapies are chemicals that patients are given. Okay. Now, these basically, unfortunately, damage DNA and also damage other healthy cells because they do not distinguish between good cells and a bad cell. Okay. Okay, so let's just talk briefly about the growth and metastasis, which is the movement, right, from one location to another location of a body. Uh, a malignant breast tumor. So you can see here the tumor starts here in the breast in one localized tissue, then basically it grows, okay, and basically it grows to more neighboring tissue, and because the breast is, has blood vessels and lymph vessels. Lymph vessels are basically what connects the immune system together. Um, these cancer cells might spread through metastasis, okay, to other parts of the body, like anywhere else, okay, and they might cause more problems as a result. Okay, metastatic tumors are treated with chemotherapeutic drugs that target the cell cycle. Okay, so uh, these drugs are targeting the cell cycle and basically um, making the cell cycle of cancer cells dysfunctional, so they do not grow anymore and they do not divide and basically die. Okay. Side effects of chemotherapy are due to the effects of the drugs on normal cells, right? So they don't distinguish between good and bad cells, as I mentioned. Okay, so there are more, research that obviously that's been going on every day to try to find better treatments for cancers. Okay, and that should end our chapter on mitosis and the cell cycle, and we will talk about the next chapter just in a bit. Okay, have a great day and see you in the next chapter. All right, bye bye now.